Alright, you ready? Cell division. You wouldn't be who you are if there wasn't for cell division. You'd still be one cell. Microscopic. This kind of shows cell division. This is in a sea urchin embryo. And it starts as one cell and it multiplies to 2. And then 4 and 8 and 16. After 16 cells, it looks like this. And then 32, 64, 128, 256, 512, 1024, 2048, 4096, 8192, 16,384, 35,000.
How?
It ends up being an organism after millions of cells. And it can become millions of cells pretty quickly because of this process we're going to learn called cell division. Now, prokaryotes do it a lot quicker than eukaryotes. In prokaryotes, we call cell division, we call it binary fission, where the cells just kind of break into, they copy their DNA first. Do you remember the DNA in a bacterium is just a single looped DNA?
Yeah.
And that's easy to copy quickly. It doesn't have a nuclear membrane around it. So bacteria, prokaryotes, can multiply rather quickly. They found some bacteria that can do it once every 8 minutes. If you can multiply once every 8 minutes, E. coli do it about every 20 minutes. You can go from one cell to millions in just a few hours. So that's why we're so concerned about infections. It's a what's called an exponential growth. One bacteria gets in your body, hours later there can be millions. Isn't that scary? People used to die from infections all the time before the invention of antibiotics, which are drugs that can kill bacteria in prokaryotes. They usually interfere with the cell wall, the making of the cell wall, because your cells don't have a cell wall. So if you took a drug that screwed up cell walls, it would only kill the bacteria inside you, not your cells. Isn't that ingenious?
Very smart.
Now, it takes a lot longer to copy one of your cells. Usually, in one of your cells, if it's fast dividing, a fast dividing cell like skin cells or cells that produce your hair, they multiply about once a day. And there's a process which we go about in copying, in multiplying the cell, and I'm going to show it to you up here on the board with my magnets. And we call this whole process the cell cycle. The cell cycle consists of several sections. G1, you remember this from regular biology? S, G2, and then prophase, prometaphase, metaphase, anaphase, telophase, cytokinesis. And if you don't remember that, well, you'll have to study it. So G1, S, and G2, we call that interphase. And most of the cell's life is spent in interphase. If this whole cell cycle takes 24 hours in a fast dividing cell, G1, S, and G2 take about 23 of those hours. And then it runs through these phases in about an hour. Again, this is prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. Now, we refer to this set right here as mitosis. Mitosis is the division of the nucleus of the cell. So we start with a nucleus. I'll draw a nucleus on the board here. Here's the nucleus. We start with a nucleus, and when we divide that into two nuclei, we're done with mitosis. This prophase, prometaphase, metaphase, anaphase, telophase, that's just the nucleus dividing into two. So we're going to do that. So we start in G1. G1 stands for the first growth phase. That's when the cell is getting bigger and bigger and bigger. It's taking in food, it's copying its organelles, it's making more endoplasmic reticulum, it's making more Golgi bodies, the little mitochondria are probably multiplying. There's just a lot going on in the cell, and the cell's getting bigger and bigger and bigger and bigger. Is there any problem with the cell being real big?
It's got too much volume.
Surface area to volume ratio issues. There's too much volume, not enough surface area. So you know what a cell's going to do in response?
Split in half.
It's going to divide in two. So, G1 is growing like crazy. Then the S phase. The S phase is when it copies its DNA. And so, here I'm showing you a cell with 6 chromosomes. Now, our cells don't have 6. You know how many our cells have?
23 pairs.
23 pairs, which is how many total?
46.
46. So this cell has 6. Our cells have 46. So you have to imagine another 40 of these spread out. There's another 40 of them in the nucleus. Now remember, this nucleus comes out at you too. So there's room in there for more of these. Okay? Now another thing you have to understand is you actually have 2 sets of chromosomes in your nucleus. You got 2 sets. You got a set you got from your mom, and you got a set you got from your dad. And they had sex.
Whoa.
Don't tell them you know. Don't tell them you know. They think you don't know. But you know.
Oh, we know.
Okay, so you got a set from your dad and a set from your mom. That's why I have a yellow set and a white set. Now, it's good that you have 2 sets because, like, on one of them you could have a gene for brown eyes, and on the other one, the homologous pair is what we call it, the matching one that you got from your dad, you might have a gene for blue eyes. You might have a brown-eyed gene from your mom and a blue-eyed gene from your dad. The 2 genes combine together to give you your phenotype, what you look like. You all remember that from regular biology?
I do.
So if you got a brown-eyed gene and a blue-eyed gene, you probably got brown eyes because brown's dominant over blue. We'll talk about that later. But you do got 2 sets of chromosomes, so we'll get into the genetics of it. Now, in S phase, all of these are copied. So we're copying the DNA. Copy, 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 copy. We copy the DNA. That takes actually about 8 hours. Because the DNA is very long, right?
Yeah.
Another thing I'm not showing you here during S phase, it's actually not coiled up into a chromosome. It's actually all spread out and diffuse in the nucleus. And the nucleus, when you look at it under the microscope, you can't see chromosomes. You just see a spread out DNA and proteins in the nucleus, and we call that chromatin. Where can I put my C words? There's a lot of C words in this chapter. Let's make a C word bank. Chromosomes. Chromosomes are actually made up of chromatin. Chromatin is DNA that's all spread out, and what it's going to do, it's going to coil up into chromosomes. So, during the S phase, it's actually all spread out. The DNA is copied, but it's not all coiled up like this. I don't have a way of showing that with magnets, but it would be cool if you could undo this magnet and make it spread out into spread out DNA. That's what chromatin is. Now, then there's a G2 phase. Oh, we got to copy the centrosomes, by the way. Do y'all remember this organelle? It's called there are little organelles called centrioles, which we learned about when we did the cell chapter. And 2 centrioles make up a centrosome. So, I don't know how well you can see these magnets, but there's 2 centrioles right here, and the whole thing's called the centrosome. And this is what's going to shoot out the spindle fibers that you might remember from regular biology. And we actually make a copy of these during S phase as well. We're going to copy the centrosomes too. So that's G1 and S. And then in G2, it's a second growth phase, and the cell might grow a little more. But during this G2 time, which is a few hours, a cell is putting together proteins that are necessary to divide the cell. There's a whole bunch of enzymes that are important if you're going to make the cell divide, and those have to be made by the endoplasmic reticulum and the Golgi bodies. And it's busy putting together all these enzymes. And then prophase comes. And prophase is when these chromosomes coil up so you can see them. Would you like to see video footage of chromosomes coiling up?
Yeah.
Okay, you would. Video footage of chromosomes coiling up. This is one of my favorite videos. I think it's actually good. Enable all, not recommended, but I do it anyway because I like to live dangerously. Hold on, let me start it over. Here's a chromosome coiling up from chromatin.
That is very nice.
Pan back. The DNA is coiling up, you see.
Whoa. She's on the screen. Where did those come
It's one long chain that coils up and forms your chromosome.
That is nice. Let's watch it again. Let's watch it again.
One more time. Here it is. That's the DNA. DNA is wound around proteins called histones. So that's DNA and protein. And it coils up. It's very orderly how it does it. There are motor molecules that pull it in, that kind of reel it in, like reeling in fishing line. You coil it up, and now you have this chromosome that's shaped like an X. So the reason why it's shaped like an X is because these chromosomes actually kind of lay over one another like that. That's why it's shaped like an X. Now don't think that because it's shaped like an X, that's the X chromosome. There's also the sex chromosomes are called the X and the Y chromosomes.
There's another one that makes a Y.
We're going to talk about those later. But every chromosome is like this. Every chromosome is kind of like this. But I'm not going to I'm not going to do all my magnets like that because they don't stick to the board as well. So we're just going to have them side by side. So every chromosome has an exact copy. These are called sister chromatids. And you can see them in the nucleus now. Even if you look with a microscope, you can see these little lines. We're going to look at these under the microscope. You may remember doing it in regular bio. You remember doing it?
Yeah.
So, this is a common diagram that you'll see on quizzes and tests. And here it shows, it's a big circle because it's a cycle. G1, the cell is growing. S is DNA synthesis. We're copying the DNA. Copy, copy. And then G2, continued cell growth and making proteins to get ready. This whole area here is interphase. Interphase is going on. I know it's pointing to interphase right there, but interphase is actually the whole thing. Big interphase, interphase. All this is interphase. And then we're going to hit mitosis, which is the division of the cell. And cytokinesis, which is when the cell breaks in two. So that's what we're doing next. I don't know why it's showing plants there. Okay, here we go. So, here are the different phases. And you can actually, the cells are under a microscope, you can see the different phases. I'll show it to you up here. And you can just kind of compare it to the pictures as we go. But prophase is when the chromosomes coil up where you can see them. And then the nuclear envelope dissolves. And by the way, you're going to have to be able to do this with the magnets. So pay attention. The nuclear envelope dissolves. The centrosomes move to opposite sides of the cell. And they're going to fire out these spindle fibers, which are protein microtubule fibers, which will shoot across the cell and attach to these chromosomes. They attach at the this portion right in the middle where the red things are. That's called the centromere. It's another C word. It's hard to keep all these C words straight. The centromere is the portion in the middle of the chromosome that captures the spindle fiber. And it's also where the two chromosomes are connected to one another. So this chromosome and that chromosome are connected at the centromere. There are actually little proteins here called kinetochores that capture the spindle fibers. Proteins in the centromere, they're called kinetochores. Write that down. It's not a C word, but kinetochores will capture the spindle fiber. And the spindle fiber is captured on this side, and the spindle fiber is also captured on this side. Comes all the way across and gets it. And once both spindle fibers have captured it, that's prometaphase. Prometaphase is when the spindle fibers capture the chromosomes. So prophase is when they coil up and the nuclear membrane dissolves and the centrosomes move to opposite sides and shoot out spindle fibers. Prometaphase is when the spindle fibers capture the chromosomes. And then, I'm not going to draw all these spindle fibers, it's too messy. You just have to imagine them. But the spindle fibers kind of drag the chromosomes to the middle. And this is called metaphase. They're all lined up in the middle of the cell. We call the middle of the cell the metaphase plate. Y'all remember this stuff? Mm-hmm. So that's metaphase. Metaphase is pretty quick. We're actually in the lab, we're going to figure out how long each one takes through looking at slides under the microscope. Figure out the length of time for each phase. And then anaphase is when these spindle fibers start pulling apart the chromosomes. The sister chromatids get separated. and move to opposite sides. They're pulled on the spindle fiber. The whole cell actually starts stretching and pulling, and the chromosomes get pulled apart. We have to do this to give each new cell a set of chromosomes. This cell will get a full 46 chromosomes. This cell will get a full 46 chromosomes. And so that's anaphase, when they separate. And then telophase, they arrive at opposite ends. These spindle fibers disappear. And the chromosomes arrive at opposite ends of the cell. And the spindle fibers disappear. And a new nuclear membrane forms. And a new nuclear membrane forms here. And now the chromosomes unwind and go back to chromatin. Do you notice we have 2 nuclei where we used to have 1? So mitosis is over. Mitosis was the division of the nucleus. We're done. And now the cell starts to pinch inward. And the cell will pinch inward from up here. We call this a cleavage furrow. It's like there's a belt. There's actually a belt that tightens. Imagine if you took a belt and you just kept tightening it and tightening it, eventually it would cut you in half, wouldn't it? There's a protein belt that forms around the cell, and it starts ratcheting. It's got little motor molecules that ratchet and tighten this belt, and it tightens and tightens and tightens and pinches this thing in two. That's what happens in animal cells. Plant cells, the belt thing won't work because the cell wall is too tough. And so what plant cells do is they just build a new cell wall between the two. So it kind of works different ways for plants and animals, but eventually this thing will separate. Bloop, you get 2 cells, and we're done with cell division. Would you like to see video footage?
Yeah.
You like to see video footage? What in the world?
An adult human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. At the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach by proteins in their centromeres called kinetochores to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles, as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells. During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately.
We'll talk about the checkpoints later. But that's pretty much it. I expect that you remember a lot of that from regular bio because we hit it pretty hard then. I need my wipers. Willis,